Cellular metabolism. In exploring cellular metabolism, we delve into the intricate network of biochemical processes that occur within living cells. Metabolism encompasses both the energy-producing pathways that generate ATP, adenosine triphosphate, and the biosynthetic pathways that synthesize cellular components. Understanding metabolic pathways is essential for comprehending how cells grow, respond to their environment, and maintain homeostasis. Below, we will examine the key components of cellular metabolism, including catabolic and anabolic pathways, the role of enzymes, and the regulation of metabolic processes. Overview of cellular metabolism. Cellular metabolism can be broadly divided into two categories. Catabolism, the set of metabolic pathways that break down molecules to generate energy. This includes the degradation of carbohydrates, fats, and proteins into simpler compounds, releasing energy stored in chemical bonds. Anabolism, the set of metabolic pathways that construct molecules from smaller units. Anabolic processes require energy input to synthesize complex macromolecules like proteins, nucleic acids, and lipids essential for cell structure and function. Catabolic pathways. Catabolic pathways primarily involve the breakdown of macromolecules into simpler compounds, accompanied by energy release. Key pathways include glycolysis. Location, cytoplasm. Process. Glycolysis is the first step in glucose metabolism, converting one molecule of glucose, 6 carbons, into 2 molecules of pyruvate, 3 carbons. Energy yield. The process generates a net gain of 2 ATP molecules and 2 NAD molecules per glucose molecule by substrate-level phosphorylation. Aerobic versus anaerobic. In the presence of oxygen, pyruvate enters the mitochondria for further oxidation. In anaerobic conditions, it can be converted into lactate, in animals, or ethanol and co, in yeast. Krebs cycle, citric acid cycle. Location, mitochondrial matrix. Process, acetyl-CoA, derived from pyruvate, enters the Krebs cycle. The cycle involves a series of enzymatic reactions that oxidize acetyl-CoA, producing NAD, FAD, and GTP, or ATP. Energy yield. Each turn of the cycle generates 3 NAD, 1 FAD, and 1 GTP/ATP. Since 2 acetyl-CoA molecules are produced from 1 glucose molecule, the cycle turns twice per glucose metabolized. Electron transport chain, etc. Location, in a mitochondrial membrane. Process. The NAD and FAD produced in glycolysis and the Krebs cycle donate electrons to the ETC, which consists of a series of protein complexes, I4, and electron carriers. As electrons pass through these complexes, protons, H, are pumped from the mitochondrial matrix into the intermembrane space, generating an electrochemical gradient, proton motive force. Chemiosmosis. Protons flow back into the matrix via ATP synthase, driving the phosphorylation of ADP to ATP, oxidative phosphorylation. The final electron acceptor is molecular oxygen, which combines with electrons and protons to form water. Energy yield. The complete oxidation of one molecule of glucose can yield approximately 30 to 32 ATP molecules, depending on the organism and efficiency of the electron transport chain. Anabolic pathways. Anabolic pathways involve the synthesis of complex molecules from simpler precursors, utilizing energy input, typically derived from ATP generated in catabolic processes. Key anabolic pathways include biosynthesis of macromolecules, protein synthesis. Amino acids are linked together by peptide bonds to form proteins. mRNA transcription and translation are essential in translating genetic information into functional proteins. Nucleotide synthesis. Nucleotides are synthesized through the de novo pathway, starting from basic building blocks like amino acids and sugars, or the salvage pathway, recycling nucleotides. Lipid synthesis. Fatty acids are synthesized from acetyl-CoA through the fatty acid synthesis pathway, leading to the production of triglycerides and phospholipids necessary for cell membranes. Photosynthesis, in plants and some bacteria. Location, chloroplasts, in plants, or thylakoid membranes, in cyanobacteria. Overview. Photosynthesis converts light energy into chemical energy stored in glucose. It occurs in two main stages, the light reactions and the Calvin cycle, light-independent reactions. Light reactions. Light energy is captured by chlorophyll and utilized to split water molecules, photolysis, releasing O and generating ATP and NADP. Calvin cycle. In the stroma, ATP and NADP are used to fix CoA into organic molecules through a series of reactions, ultimately producing glucose. Role of enzymes in metabolism. Enzymes are biological catalysts that facilitate biochemical reactions by lowering the activation energy required. Key features of enzymes include specificity. Enzymes are highly specific for their substrates, allowing for precise regulation of metabolic pathways. Cofactors and coenzymes. Many enzymes require cofactors, metal ions, or coenzymes, organic molecules, often derived from vitamins, for activity. For example, NAD serves as an electron carrier in redox reactions. Regulation. Enzymatic activity can be regulated through various mechanisms, including allosteric regulation, binding of molecules at sites other than the active site, competitive inhibition, substrate mimics blocking the active site, and feedback inhibition, end products inhibiting earlier enzymes in the pathway. Regulation of metabolism. Metabolic pathways are tightly regulated to maintain cellular homeostasis and respond to environmental changes. Key regulatory mechanisms include allosteric regulation. Enzymes may have sites where molecules bind and induce conformational changes, activating or inhibiting enzyme activity. Hormonal regulation. Hormones such as insulin and glucagon play crucial roles in regulating metabolism by promoting or inhibiting specific metabolic pathways. For example, insulin promotes glucose uptake and glycogen synthesis, while glucagon triggers glycogenolysis and gluconeogenesis. Energy charge. The energy state of the cell, often reflected by the ATP/ADP ratio, influences key metabolic processes. High ATP levels promote anabolic processes, while low ATP levels activate catabolic pathways. Genetic regulation. The expression levels of genes encoding metabolic enzymes can change in response to cellular conditions, impacting the overall metabolic capacity of the cell. Integration of metabolism. Metabolism does not occur in isolation. Different pathways intersect and communicate with one another. Metabolic pathway interconnectivity. Catabolic and anabolic pathways are interconnected through key intermediates. For instance, carbohydrates, fats, and proteins can all yield acetyl-CoA, which feeds into the Krebs cycle. Nutrient sensing. Cells continuously monitor nutrient levels and energy states, through pathways that sense glucose, amino acids, or fatty acids, adjusting metabolic activities accordingly. Cellular signaling. Communication between cells can also impact metabolism. For example, during fasting or exercise, signaling molecules coordinate metabolic shifts to ensure that energy is available when needed.